define themselves. The embryonic stem cells will become tissue of everything in the lump. The adult stem cells will become cells of the tissue in which it finds itself. So when we talk about health, obviously adult stem cells suddenly become the real thing. This is where all the promise is in terms of health, because you want cells that become cells of the tissue in which they migrate. All this concept can be summarized essentially in this brief concept here, in this animation. So this represents essentially the blood circulation, and we have the bone marrow. It's obviously a schematic representation. When you have something happening in the body, the area that has the problem is going to start to release specific chemicals that have been well identified in science called granulocyte colony stimulating factor, GCSF. Forget about these terms, they're not important. But there's a compound very specific that is released. It migrates to the bone marrow, and when it reaches the bone marrow, it's going to start to trigger the release of stem cells from the bone marrow. Now these stem cells, when they're released, they don't know where to go. They're circulating everywhere in the body. They go everywhere. Sometimes I use this analogy. It's a little bit like if I take an apple pie fresh out of the oven and I put it somewhere here in the building and I tell you, go find it. What do? You don't know where it is when you start. You smell around, and when you walk in front of a room where the apple pie is, you stop and you say, that's here. And you enter into the room. Stem cells do the same thing. They circulate, and when they arrive in the tissue that has the problem, that tissue is releasing a very specific compound. Think of it as the apple smell, called stromal-derived factor 1, SDF1. When that compound touches the stem cell, or reaches your nose, the stem cell turns around, and the mechanism here is that the moment that the molecule touches the stem cell immediately expresses adhesion molecule. So the stem cell is going to cling to the bone marrow, to the capillary, stop in the blood flow, and then will migrate in the tissue. And when it comes in contact with cells of that tissue, it is going to slowly become cells of that tissue, sort of replacing the tissue that is having the problem and bringing back optimal function in the tissue. That's what stem cells are doing in the body. Isn't it fascinating? It's an amazing process. And what I'm describing here in this drawing is taking place in the liver. The same thing is taking place in the brain. The same thing is taking place in the kidney. Same thing is taking place in the muscles. So it's essentially almost anything that they look at, they can find that the stem cells can become cells of that tissue. Here's a study where scientists took stem cells and they put them in a beaker on a mesh. The mesh was, so the stem cells here are in yellow, those yellow dots, put on a mesh that has holes that are big enough to allow molecules to go through, but small enough to prevent cells to go through. Okay, so you put these stem cells at the top, and at the bottom, they put a piece of crushed liver. So if you end up with cells of the liver at the top, it is not because cells of the liver migrated on top of the screen. They cannot cross that layer. This is a screen, it's a filter. It prevents cells from going through. When they looked at these cells on this mesh, 8 hours later, they were morphing, transforming themselves into liver cells. Now, understand why it took so long for us to see this phenomenon. It's because it is invisible. If I use the traditional mean of looking at a tissue, I take a piece of tissue, I slice it in a thin slice, I put it in a microscopic slide, and I look at it. What do I see? Liver cells. It is absolutely impossible to tell that yesterday that liver cell was a stem cell. It's impossible unless I can use something to tag it. And that leads us back to where I started, green fluorescent protein. Extracted from a deep ocean jellyfish, it is a protein that is spontaneously fluorescent. Because it is spontaneously fluorescent and because it's a protein, it's very easy. I mean, I'm making that easy. We're talking that Nobel Prize, 2008 Nobel Prize in Chemistry, was on a span of about 40 years of research. I'm making it look like it's nothing. But in theory, it's a protein, so it's easy to derive the DNA that encodes for that protein. Once you have the DNA, it's easy to inject it in a cell, in a stem cell. Now go back to what we were talking about just before. If I take now an adult stem cell that has that fluorescent protein in it, and I inject it in the skin, what do I get? I was looking to get a teratoma before, and I was getting nothing, so the conclusion is that they're not good stem cells. I get a patch of fluorescent skin. So the revelation here is that stem cells have the same adult stem cells, same ability, but the plus is that instead of becoming everything, they only become cells of the tissue in which they are. Okay? So, and here's what cells will look like when they do glow with the presence of that protein. So what normally would be invisible becomes visible. So it's the tracking device that we use to see where whales are going. It's a tracking device to see that the stem cells are going into these different tissues and becoming cells of these tissues. Here's how all of this is sort of coming together in a few studies. The study of Orlik, that was done at the NIH. So I'm using studies here that are extremely strong, coming from very credible scientific teams. So Orlik, what he did was very simple. He irradiated mice, which kills all stem cells. And then I inject new stem cells, but with that protein, green fluorescent protein. So you have green fluorescent stem cells. And then I trigger a heart problem, lack of oxygen in the heart, which leads to the death of part of the heart tissue by tying up the coronary artery. And then they came back and they looked at the animal 2 weeks later. And what they could see is that the portion of the heart that was affected was now fluorescent green. Meaning that these cells that are now heart cells today, 2 weeks after, they came from the bone marrow. But what is beautiful in this is that they reach the area and they become cells of the heart on their own. It's a beautiful picture of this phenomenon. What we show here is the picture of a muscle that was totally destroyed or killed on the first day of the study, and then we add the same protocol here with mice with fluorescent stem cells, and these cells on their own migrated in the muscle, and you can look, the muscle has been totally reconstituted, entirely green. It's a great picture to show here what we're talking about. The moment we have that tool, it becomes almost like child play to reveal what stem cells are doing in the body. So now, the number of stem cells. What would happen if I put more stem cells in circulation? What happens in the power of stem cells like this to repair various tissues? So the same kind of studies. I use two groups of mice. I trigger a death of the cardiac tissue by tying up the coronary artery. I split the mice in two groups. One group is control. I do nothing. The other group, I inject in the animals GCSF. Remember that compound that triggers stem cell release. So the two groups, the only difference, one group there's nothing. The other group, I have more stem cells in circulation for a few days. This was done for about 8 to 10 days. 27 days later, when they looked at the control animals, you can see that in the heart there was a lot of scar tissue. This is the blue that you can see. Lots of scar tissue, no new blood vessels, 17% survival, so 83% fatality, and then very, very compromised cardiovascular function. This is when nothing is done. In the animal who had more stem cells circulating in their blood, in the same amount of time, we're talking about roughly a month, total renewal of the ventricular wall, no scar tissue, new functional blood vessels tying up in the other ventricles, 73% survival, and quasi-normalized cardiovascular function. That study is telling us that in a month, there is a way, in a month, to reverse the consequence or revive a part of dead heart tissue. This is totally amazing. The reason why this protocol is not used as such in humans, it's because what is done here in mice gives severe, severe side effects in human. If we use GCSF for a little bit more, too long, more than 5-6 days, the consequences are heart attack, stroke, fairly severe side effects. So the idea of telling you, well, to help you from your first problem, we're going to give you, a heart attack. It's not a good trade-off. It's not a great marketing message. So these compounds are not ready to go to market, but they demonstrate a phenomenon that is very, very interesting. Now, GCSF is a powerful, powerful mobilizer of stem cells, meaning it triggers a vast release, a huge release of stem cells from the bone marrow. StemEnhancer that we'll talk about does not do that. So I do not want here to tell you that StemEnhancer will do this. What I want to do here by showing this is to show the power of stem cells, that if you really have a lot circulating in the blood, what you can end up in terms of repair is pretty phenomenal. But the key issue in here is the number of stem cells circulating in the blood. And that was captured by a study published in 2005. And now many studies have come out and have shown the same kind of results or the same kind of information, which is more stem cells in the blood equates to greater health. So if you have a little bit more stem cells every day, you simply allow the body to renew itself every day. So it's not about healing. It's not about anything phenomenal. It's a little bit like if I'm telling you, I am going to give you a month's supply in one day of food.